Okay, we're on to step 2. And if you look at the picture, it's endocytosis. Endocytosis is the process where cells bring really big things inside through vesicles. And a vesicle is just the little pocket that envelops the virus, and the pocket itself has its own membrane. So, number 2, the cell brings in the virus by endocytosis. Step 3, uncoating. So the virus uncoats, releasing its 8 segments of RNA and its polymerase into the cell. Step 4, nuclear import. The virus segments and polymerase go into the nucleus. Step 5, the RNA is replicated for progeny virions. So I might make a separate video of this, but basically the virus's polymerase takes the negative sense RNA, makes a positive sense copy, and then it makes a negative sense copy to make more viral genomes. And remember, it's got 8 separate segments of RNA, so it's going to have to do, and we're making a lot more than just one virus, so it's going to be copying a lot of RNA. All right, step 6, transcription. That's still in the nucleus. Viral mRNA is transcribed and translated to make more H and N proteins and polymerase. Now, the virus does have more proteins, and it is more complicated than that, but if you ever take virology in college, you may learn more about it. But for our purposes, that's good enough. So, step 7, nuclear export. The viral RNAs are exported out of the nucleus. Step 8 is assembly. So they assemble with the H and N proteins and the polymerase at the cell membrane. And then step 9, budding. The progeny viruses bud off of the membrane and are released. And this whole cycle takes about 6 to 8 hours. And eventually, after enough viruses are made and they're budding from the cell, it will kill the host cell, which is why you get so sick. One of the reasons why you get so sick. Okay, I've got another video. This gives you a little bit more detail of influenza replication. It's not covered on the test, but if you find this interesting, by all means, watch this video. Okay, antigenic drift. This was covered in my podcast, Will Kill You. So there's 2 ways that that gives rise to so many strains of viruses, antigenic drift and antigenic shift. And I'm going to cover antigenic drift first. So as an RNA virus, influenza mutates quickly, which can cause changes to the H and N glycoproteins, or antigens. These changes make it harder for our immune system to recognize and fight the virus. This is called antigenic drift, and it's a small change. So I want you to think of drifting down a river, and the course you are taking is changing slowly, small changes if you're just drifting down a river. Okay? Think of drift as a small change like drifting down the river. Watch that video, please. And then antigenic shift. So if you are changing course in a river, it's a big change, right? Like you change course to a new stream or a new river. So think of shift as a big event. It's rare. And this is when an animal gets infected with 2 different strains of flu at the same time. So because RNA has 8 segments of RNA, it can mix and match those RNAs from, say, a chicken and a human, and the human viral RNAs and the chicken viral RNAs mix and match and create a brand new virus. This is a big change. This is believed to what made the 1918 flu pandemic so deadly, so that a pig got infected from a human and a bird at the same time, and the human and bird mixed RNAs and made a new virus. So, like I said, it's a big change, and think of it like shifting course in a river. And then there's a video on antigenic shift, and this shows the difference between antigenic shift and antigenic drift. Antigenic drift is just a small change in the N or H proteins, whereas antigenic shift is mixing of RNAs from two separate viruses.